Immunology Insights, Basics and Beyond, Chapter 68, CRISPR and Gene Editing in Immunotherapy. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our previous chapter, we explored nanotechnology in immunotherapy, how engineered particles can protect fragile payloads, improving targeting and make immune modulation more precise by controlling where and how drugs are delivered. Today, we move from delivery to something even more fundamental, changing the biological wiring inside cells. In this chapter, we will look at how CRISPR and gene editing are used to redesign immune cells, tune immune pathways, and build therapies that are more durable, more specific, and more scalable than what was possible with drugs alone. So let's talk science. What do we actually mean by gene editing in immunotherapy? Gene editing means making targeted changes to DNA in living cells. In immunotherapy, this is usually done to either improve immune cells used as therapies, remove biological breaks that limit immune function, or to discover new targets by systematically testing which genes matter most for immune responses. A key point is that gene editing is not only about switching genes off. There are two main categories. First, knockout, disabling a gene to remove a function, for example, removing an inhibitory receptor pathway. And second, knock-in, inserting or rewriting DNA to add a new function, for example, inserting a CAR into a defined genomic site or introducing a protective safety switch. The promise is clear. Instead of only giving a drug that temporarily blocks a pathway, gene editing can sometimes create a durable, programmable change, especially in cell-based therapies. Now let's look at the core concept of CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and originates from a bacterial defense system against viruses. In biotechnology, it has been adapted into a programmable gene editing tool. The simplest CRISPR system has two key parts. First, a Cas enzyme, often Cas9, that can cut DNA. Second, a guide RNA that brings Cas to a specific DNA target by base pair matching. When Cas9 makes a targeted cut, the cell repairs the DNA using its own repair machinery. Two repair modes are especially important. First, NHEJ, or non-homologous end joining, a fast repair that often introduces small insertions or deletions. This is why it is so useful for knockouts, for example, knocking out PD-1 signaling by disrupting the PDCD1 gene. And second, HDR, homology-directed repair, a more precise repair pathway that can copy a provided DNA template. This is one route to knock-ins, such as inserting a therapeutic receptor sequence at a chosen genomic location. So, how do knock-ins work in practice? Conceptually, you provide the cell with an instruction template that contains the desired sequence flanked by matching DNA regions, and the cell can copy that sequence into the cut site during repair. Because HDR is most active in dividing cells, it can be less efficient in some settings. So researchers also explore alternatives. One of these is HITI or HITI, which means homology-independent targeted integration, which aims to harness NHEJ-like repair to integrate a DNA payload at a defined cut site, making integration more feasible in cells that are not actively dividing. Finally, there is the practical question of how the CRISPR machinery is introduced into cells. In immunotherapy, this is often done ex vivo, so outside the body, using transient delivery formats such as Cas protein complexes or Cas encoded as RNA, so that the editing tool is present only briefly. In some settings, viral vectors are used as delivery vehicles for larger DNA templates or transgenes. The exact choice depends on whether you want a transient edit, a stable inserted gene, or both. So why does CRISPR fit immunotherapy so well? Well, immune cells are naturally built to detect and destroy threats, but in disease they face predictable limitations. Tumors suppress them, chronic infections exhaust them, and in autoimmunity the opposite problem occurs. Immune responses are too strong or misdirected. CRISPR fits particularly well because it can address these bottlenecks at the level of cell programming. First, removing inhibitory brakes that drive exhaustion, such as PD-1 linked pathways. Second, improving persistence by tuning survival and differentiation programs, for example, favoring long-lived memory-like states. Third, making allogeneic products safer by reducing graft versus host mechanism. And fourth, adding control features like safety switches that can stop the therapy if severe toxicity occurs. In other words, CRISPR can make immune cells not only stronger, but also more predictable and more controllable, crucial for real-world clinical use. Now let's look at ex vivo editing, so reprogramming immune cells outside the body. The most mature applications of CRISPR in immunotherapy are edited immune cell products, especially T cells and increasingly NK cells. Examples include: first, making allogeneic cell therapies safer and more universal. Autologous therapies use a patient's own cells but are slow and individualized. Allogeneic therapies use donor cells and can be manufactured in batches, but they bring often two risks: graft versus host disease and host rejection. CRISPR can help by reducing the activity of the endogenous T cell receptor system that drives graft versus host reactions in donor cells. Additional edits can reduce immune recognition of the donor cells, which is one way researchers aim to extend persistence of off-the-shelf products in recipients. Second, making engineered cells harder to suppress. Tumors frequently suppress T cells through inhibitory ligands and chronic stimulation. One concrete strategy is to knock out genes associated with exhaustion signaling or inhibitory receptor pathways, so engineered cells maintain cytotoxic function longer in hostile tumor microenvironments. This concept is being explored in both CAR T cells and TCR engineered T cells. Third, more predictable engineering through targeted knock-in. Beyond random insertion of a transgene, CRISPR can be used to place a therapeutic receptor into a defined genomic locus. A common goal is uniform, regulated expression, reducing variability from cell to cell and potentially lowering risks linked to uncontrolled expression. In principle, inserting a CAR into a defined T-cell receptor locus can both standardize expression and simultaneously reduce endogenous receptor activity. And fourth, engineering NK cells. NK cells are attractive because they can be used allogeneically with lower graft versus host risk and have MHC-independent recognition features. Gene editing is being explored to improve NK persistence, for example by adding supportive cytokine signaling programs, and to increase resistance to suppressed tumor environments, sometimes combined with CAR constructs to create CAR-NK products. Now let's also briefly look at in vivo editing, so reprogramming cells inside the body. In vivo gene editing aims to deliver CRISPR components directly into a patient so the desired edit happens inside target cells in their natural tissue context. Conceptually, this could reduce manufacturing time and cost compared with ex vivo cell therapy and could make immune engineering available without harvesting or expanding cells first. However, it is harder to control. The key challenges are hitting the right cell types and only those, limiting duration of CRISPR activity, and avoiding unintended edits or inflammatory reactions. For immunotherapy, in vivo editing is therefore still earlier stage than ex vivo approaches, but it represents a logical future direction, especially if delivery system and cell type targeting become more reliably precise. Now let's look at some application examples of CRISPR in a disease context. starting with CRISPR in cancer immunotherapy. So what does it enable? In cancer, the central question is often how do we keep immune cells active inside a tumor long enough to win? CRISPR supports several directions. For example, first, resistance to checkpoint-driven exhaustion by editing pathways that limit sustained T-cell function. Second, improve performance in solid tumors by tuning programs that influence trafficking, persistence, and survival under hypoxia and nutrient limitation. And third, discovery of new targets. Large CRISPR screens can systematically identify which tumor genes drive immune evasion or which immune cell genes correlate with durable killing versus early dysfunction. That directly feeds the development of next generation checkpoint targets and rational combination strategies. Now let's look at application examples of CRISPR beyond cancer. So, autoimmunity, infection, and transplantation. Cancer is not the only place where immune programming matters. First example is autoimmunity. In severe refractory autoimmune diseases, researchers are exploring whether deep immune resets can be achieved by eliminating pathogenic immune compartments and allowing reconstitution. In parallel, regulatory T-cell therapies aim to restore tolerance rather than destroy immunity. Gene editing could help to improve stability and specificity of tolerogenic cell products and by adding safety controls. Second, in infectious diseases. Chronic infections such as HIV can drive long-term immune exhaustion and functional impairment. Gene editing has been explored as a tool to enhance antiviral immune cells or to modify host factors relevant for infection control. Here, the goal is targeted and durable control without triggering harmful inflammation, an area where careful balance is essential. And third, transplantation. The same principles that enable safer allogeneic cell products also apply to tolerance strategies. Engineering regulatory cells that suppress alloimmunity or creating standardized immune cell sources with reduced immunogenicity could reduce the need for long-time broad immunosuppression. But there are also newer editing modes coming up, so the use of CRISPR beyond DNA cutting. Not all CRISPR tools rely on classical double-strand breaks. Two important next-generation concepts are: first, base editing. So it converts one DNA base into another without cutting both strands, useful for precise single-letter changes. Second, prime editing. That can write defined small edits using a guided search and replace mechanism, potentially expanding what can be edited with fewer large breaks. A related idea is that Cas9, which binds DNA but does not cut it. By fusing it to regulatory domains, it can turn genes up or down, offering a way to tune immune programs without permanently rewriting DNA sequences. But of course, there are safety concerns, there are limitations, and it's not just plug and play. CRISPR is powerful, but clinical gene editing demands strict safety discipline. Key issues include: first, off-target edits, unintended cuts at similar sequences, which is why extensive sequencing-based quality control is essential. Second, on-target complexity. Even intended edits can sometimes lead to larger deletions or rearrangements than expected. Third, cell stress and fitness. DNA damage responses can reduce viability or alter the functional state of therapeutic cells. Fourth, immunogenicity. Cas proteins are foreign. Immune recognition becomes especially relevant for in vivo approaches. And fifth, delivery and control. Ex vivo editing is more controllable. In vivo editing faces major challenges because tissue targeting and duration of exposure strongly affect safety. These constraints shape why most clinical immunotherapy applications today favor controlled ex vivo manufacturing. Now let's have a brief look at the clinical relevance. So what does CRISPR change in practice? The practical contribution of CRISPR to immunotherapy is not one single product. It is a capability shift. It makes it increasingly feasible to build immune therapies that are more standardized, so batch manufactured allogeneic options, more durable, so resistant to suppression, more precise, so defined genetic programs, and potentially safer, so with control mechanisms and better manufacturing consistency. This is why CRISPR belongs into a future directions block. It is becoming and enabling technology across oncology, transplantation tolerance strategies, and immune dysregulation diseases. To conclude, CRISPR and gene editing add a new dimension to immunotherapy. We are no longer limited to blocking pathways temporarily. We can reprogram immune cells at the level of their instruction set. By combining knockouts that remove breaks, knock-ins that add new functions or improve control, and newer editing modes that reduce reliance on DNA breaks, the field is building immune therapies that are increasingly programmable, scalable, and precise. In our next chapter, we will step back into the human story behind this platform and meet one of its key pioneers, Jennifer Doudna, and the development of CRISPR-Cas9, how a discovery rooted in microbial biology became a technology reshaping modern medicine. Before closing, I have a small request. If you enjoyed my content and want to hear more, please like and subscribe. It's a small step for you, but it helps me a lot. So thank you for listening and see you next time.